Welcome to this exploration of how psychedelics affect the brain and consciousness. We'll examine the scientific principles behind this fascinating area of research, conducted under strict medical and ethical supervision. First, let's define what we're talking about. Psychedelics are a class of compounds that produce profound changes in perception, mood, and cognition. The classic psychedelics include LSD, psilocybin, which is found in certain mushrooms, DMT, and mescaline. These all share a similar chemical structure to serotonin. These compounds primarily work by binding to serotonin 2A receptors in the brain, particularly in the cortex. These green dots represent 5-HT2A receptors distributed across the cortex. When psychedelics bind to these receptors, they trigger a cascade of neural events. To understand what psychedelics do, we first need to understand a key brain network called the default mode network, or DMN. The default mode network is most active when you're at rest, mind-wandering, or thinking about yours. It includes regions like the medial prefrontal cortex and posterior cingulate cortex. Think of the DMN as your brain's autopilot mode. It maintains your sense of self, your narrative identity, and it filters sensory information based on prior experiences. Here's a simplified representation. The default mode network has high connectivity and activity when you're doing nothing in particular. It's constantly chattering away. So what happens when someone takes a psychedelic under controlled conditions? This is where things get really interesting. The most robust finding from brain imaging studies is that psychedelics cause a dramatic decrease in default mode network activity and connectivity. Studies using functional MRI show that DMN activity can decrease by 30 to 50% during peak psychedelic effects. The default mode network quiets down. The normal chatter and filtering that happens constantly is reduced. This correlates strongly with the subjective experience of ego dissolution. Now you might ask, if the default mode network shuts down, what's happening instead? Great question. What researchers observe is a dramatic increase in communication between brain networks that normally don't talk to each other much. This phenomenon is called increased global brain connectivity, or brain network desegregation. Brain regions that usually operate independently start coordinating. Imagine different brain networks as separate cities. Normally they operate somewhat independently. Under psychedelics, it's like building highways between all the cities. Visual and auditory systems crosstalk. Memories blend with emotions in unusual ways. One of the leading theoretical frameworks for understanding this is called the entropy model, developed by Robin Carhart Harris and colleagues. Entropy is a concept borrowed from physics and information theory. It measures disorder or randomness in a system. This is the Shannon entropy formula. H is entropy, P sub I is the probability of state I, and we sum over all possible states. In normal waking consciousness, your brain operates in a relatively low entropy state. Neural activity is somewhat predictable and constrained by the default mode network. Under psychedelics, brain activity becomes higher entropy. Neural patterns become more diverse, less constrained, and less predictable. Look at this visualization. The normal state has neural activity clustered in a narrow range, very organized. The psychedelic state shows much more spread out, diverse patterns of activity. Let me show you a rough calculation to quantify this. Let's estimate how much information processing capacity increases. The human brain has approximately 100 billion neurons, that's 10 to the 11 neurons. In the normal waking state, let's say about 10% of these neurons are actively participating in coordinated patterns at any moment. That gives us 10 to the 10 neurons, or 10 billion neurons in coordinated activity. Under psychedelics, with increased global connectivity, let's estimate this increases to 15% of neurons participating. That's 1.5 times 10 to the 10 neurons, a 50% increase in the number of neurons participating in coordinated patterns. The number of possible information states grows exponentially with n, the number of active units. It's proportional to 2 to the power of n. So the ratio is 2 to the 1.5 times 10 to the 10 over 2 to the 10 to the 10. This is astronomically larger, showing the massive increase in possible brain states. This back-of-envelope calculation helps explain why psychedelic experiences feel so incredibly rich and complex. The brain is exploring a vastly expanded state space. Now let's talk about the therapeutic potential. One of the most promising areas is treatment-resistant depression. Depression is often characterized by rigid, repetitive, negative thought patterns. Patients get stuck in loops of rumination. The hypothesis is that psychedelics help reset these rigid patterns by temporarily disrupting the default mode network and allowing new patterns to form. On the left, we see rigid loops representing depressive rumination. On the right, after psychedelic therapy, we see more flexible, diverse neural patterns. Clinical trials show impressive results. In one study at Johns Hopkins, about 70% of participants showed significant reduction in depression scores. Remission rates, where depression essentially goes away, are around 50 to 60% in some studies. This is remarkable compared to traditional antidepressants. Importantly, these aren't just temporary effects while the drug is active. Benefits can last weeks to months after a single session. How can a drug that's out of your system in hours produce effects lasting months? This brings us to neuroplasticity. Psychedelics appear to promote neuroplasticity, the brain's ability to form new connections. They increase dendritic branching and spine density. One mechanism is through increased BDNF, brain-derived neurotrophic factor. This is like fertilizer for neurons, promoting growth and survival. Studies in animal models show BDNF levels can increase by 200 to 300% in certain brain regions after psychedelic administration. This neuron is forming new connections, new branches, new synapses. This structural change could explain the lasting therapeutic effects. There's another fascinating hypothesis called the critical period hypothesis, proposed by Gould-Dolan and colleagues. The idea is that psychedelics temporarily reopen critical periods, windows of time during development, when the brain is especially plastic and ready to learn. It's as if the adult brain briefly regains some of the learning capacity of a child's brain. This could allow for rapid therapeutic change. Interestingly, different psychedelics open these windows for different durations. In animal studies, psilocybin reopens critical periods for about 2 weeks. The critical window might last 2 to 4 weeks in humans. This is when therapeutic interventions might be most effective. Let's shift to creativity. There's growing interest in how psychedelics might enhance creative thinking. There are many anecdotal reports of breakthroughs from notable figures. Francis Crick reportedly used LSD. Kary Mullis, who invented PCR, credited psychedelics with expanding his thinking. Creativity involves divergent thinking, the ability to generate many diverse, novel ideas. This is different from convergent thinking, which focuses on finding one correct answer. Divergent thinking radiates out in many directions from a central problem. Psychedelics may enhance this by increasing the diversity of neural states explored. Now you might ask, what does the actual scientific data show about creativity? This is where we need to be careful. During the acute psychedelic state, when people are actually high, creativity tests show mixed results. Some measures improve, others worsen. Interestingly, the clearest improvements in creativity measures appear 1 to 2 days after the experience, not during it. This suggests there's a sweet spot, a window after the acute effects, where increased neuroplasticity and openness combine with normal cognitive function. Now we must discuss the risks and what we don't know. This is critical for scientific honesty. Psychedelics can produce intensely challenging psychological experiences. Anxiety, paranoia, and overwhelming emotions can occur, especially without proper support. There's evidence that psychedelics can trigger psychosis in individuals with pre-existing vulnerability, such as those with family history of schizophrenia. The risk in the general population is estimated at 0.5 to 1%, but it's much higher, perhaps 10 to 20% in those with family history of psychotic disorders. We don't know the long-term effects of repeated use on brain structure. Most studies have looked at single or few doses in controlled settings. The concepts of set and setting are crucial. Set refers to mindset and expectations. Setting refers to the physical and social environment. Both dramatically influence outcomes. In therapeutic contexts, these are administered in carefully controlled environments with trained facilitators. This is completely different from recreational use. Let me summarize what we know versus what remains unknown. Here's what we know with reasonable confidence. Psychedelics reliably decrease default mode network activity and increase global brain connectivity. They promote neuroplasticity in animal models through mechanisms like increased BDNF. Clinical trials show genuine promise for treatment-resistant depression, PTSD, and anxiety in terminal illness. Therapeutic effects can persist for weeks to months from relatively brief interventions. But here's what we don't know. We don't know the optimal dosing schedules and frequencies for different psychiatric conditions. Long-term safety data with repeated use over years is lacking. We can't predict who will respond positively versus who might have negative outcomes. The exact mechanisms by which brief experiences produce lasting change are still debated. Is it neuroplasticity? Psychological insight? Both? How different psychedelic compounds compare? Is psilocybin better than LSD for certain conditions? We don't have enough comparative data. Let's zoom out to the bigger picture. Psychedelic research is opening new windows into consciousness. Psychedelics provide neuroscientists with a tool to systematically perturb consciousness and observe the neural correlates. By observing what happens when the sense of self dissolves, we learn about the neural basis of selfhood and ego. These substances challenge our assumptions about what normal waking consciousness is. Is it the optimal state, or just one point in a larger space? Imagine the state space of consciousness. Normal waking consciousness is just one point. Psychedelics explore other regions. Meditation explores yet others. This helps us map the full landscape. Let's do one more calculation using a concept from integrated information theory. Integrated information theory proposes that consciousness arises from integrated information. The theory uses a measure called phi. Phi measures how much a system is more than the sum of its parts, how much the whole integrates information beyond what parts do independently. If psychedelics increase global connectivity, we'd expect phi to increase. Let's estimate this. As a rough approximation, let's assume phi is proportional to the number of connections squared. Phi is proportional to n squared, where n is the number of effective connections. If global connectivity increases by 50%, n becomes 1.5 times n. The new phi equals 1.5 n quantity squared, which is 2.25 times n squared. That's 2.25 times the normal phi. This predicts a 125% increase in integrated information. This could correspond to the intensely conscious, hyper-aware quality of psychedelic states. Before we conclude, let's address the ethical dimensions of this research. All research must follow strict ethical protocols. Informed consent is paramount. Participants must understand risks and benefits. Rigorous screening for psychological and medical contraindications is essential. Not everyone is a suitable candidate. Trained facilitators and medical oversight are required throughout. This isn't something people should try to self-administer. Integration support after sessions is crucial. The experience itself isn't enough. People need help making sense of it and applying insights. Many of these compounds have been used for centuries in indigenous contexts. Research must respect these traditions and not appropriate them without acknowledgement. Let's conclude by looking at where this field is heading. Psychedelics are proving to be powerful tools for understanding brain function and consciousness. The therapeutic potential is real, but it requires much more careful study before these become mainstream treatments. These are not panaceas. They work for some people, but not everyone. We need to understand who benefits most. Research into mechanisms is accelerating. Technologies like high-density EEG and advanced neuroimaging are providing unprecedented detail. Regulatory pathways are opening. The FDA has granted breakthrough therapy designation for psilocybin-assisted therapy for depression. Many questions remain about long-term effects, optimal protocols, and how to scale these therapies responsibly. Let me leave you with a final thought. These compounds are revealing that consciousness is more flexible and multifaceted than we typically experience. They show the brain can operate in radically different modes, challenging our understanding of what's possible. This research bridges neuroscience, psychology, and philosophy in unique ways. And it reminds us how much we still don't understand about the nature of mind and subjective experience. The science is rigorous, exciting, and still very much unfolding. We're at the beginning of what may be a transformative era in psychiatry and consciousness research.